Hello and welcome to this video on cloning, the science fiction trope of choice in many settings. Cloning is generally accepted to be a copy of one living being in another. That being said, not all cloning is the same, and this is only part of the definition of cloning. It describes a variety of different things that are all put under the heading of cloning. The thing they have in common is that they all produce genetically identical copies of a biological entity. There are three main types: molecular cloning, organism cloning, and therapeutic cloning. Of these, the first two are common, while the third is relatively rare until very recently. Molecular cloning involves taking a relatively small DNA sequence, generally one gene or promoters and similar other modifying parts of a gene, and creating multiple copies of it. You then have the more advanced and, frankly, most difficult of all these, organism cloning. This is probably what you're imagining when you think of cloning. This involves the creation of an identical copy of the entire organism. Sometimes organism cloning is also called reproductive cloning, and we'll explain why that is shortly. Finally, there's therapeutic cloning, and this is both a relatively new thing and considerably more controversial. It involves the cloning of a human embryo, and that's because the human embryo will produce stem cells that can be turned into anything. This means that they're a great asset for trying to treat diseases that otherwise have no treatment. The process of doing this does destroy the developing embryo. Another way to look at this is that there is naturally occurring cloning and artificial or unnatural cloning. We'll start with natural cloning as they're probably the best place to begin and the one that people are most familiar with. There are two examples. One of these you might be more familiar with: twins. Twins are people who start as a single fertilized embryo. This eventually splits to create two sets of cells that develop along the same lines to create two fetuses. Both fetuses have the same genome as they started from the same material. It's like making two copies of a document instead of one. The other example is bacteria. By reproducing asexually, the bacterium passes on two exact copies of exactly the same genome. This occurs in both daughter cells that are the offspring. This is the same genes, structure, and function of the original bacteria, just in two new cells. These two examples highlight the big differences in clones and clone purposes. One is a single cell, while the other is a whole organism. The ability to clone a single cell, like in the case of bacteria, is much easier than a whole creature. This is where artificial cloning comes into play. The problem we have with cloning a whole organism is that there is a lot of genetic material that needs to be copied over amongst everything else. This problem is evidenced by the first cloned organism, Dolly the sheep. They were developed in 1996, and it's worth noting that although Dolly is viewed as a landmark achievement, she was one of 276 embryos they tried this with. One of 276. That is a tiny, tiny fraction of a percentage of success. Dolly was cloned from its mother and unfortunately beset by many illnesses that could be traced back to genetic issues. The way this was done was they took the egg from the mother sheep and took a set of cells from the mother sheep. These cells had their genome within them taken out, that is all the nucleic material or DNA. This was then put into the egg with the egg DNA being taken out. This left two complete sets of chromosomes within the egg, and so the egg was effectively fertilized. This meant that it could start to grow. This leads to the kinds of cloning that could be called unnatural or artificial. It's the kind of technology used to create copies of genes used for genetic modification. Gene cloning produces copies of genes or segments of DNA. And this is incredibly important if you're trying to be able to, say, modify the genome of a crop to make it more productive. remove illnesses from people, and more. In recent years, this particular area has been of greater interest, especially after the FDA approved various crops, meat, and milk products from cloned animals in 2008. They effectively decided that the meat, milk, and similar from these animals was the same as that from non-cloned animals. This has opened up huge pathways for companies now looking to try and create meatless meat. That is, meat that isn't derived from an animal. The Impossible Burger, for instance, and similar. All of these products rely on the ability to clone cells, have them proliferate, and turn into something like a meat product. It allows you to take cells from an animal and never have to harm the animal again, and yet create meat in a test tube. The problem with just about every clone you're going to find is that they will inevitably observe adverse health effects. This is more commonly seen in large organisms where complete and complex genomes are being cloned. As we mentioned in Dolly the sheep, she was just one of 276, the only successful clone. When we're looking at other examples of cloning, there have been issues around the size at birth, defects in vital organs, damage to the liver, brain, heart, premature aging, and more. All of these can be traced back to how you create the clone. The original cell is going to be using somatic cell DNA, that is, cells in the body that aren't normally involved in any kind of sexual reproduction. Things like your muscle cells, skin cells, all of these are somatic. They will age, and the genome within them will age. This is observed in the shortening telomere, or the cap at the end of DNA. When it gets to a certain length, the cell can no longer divide, and as a result, the cell will die. When this happens, other cells will come along and replace it. By creating a clone using somatic cells, you artificially shorten the lifespan of the organism. That assumes that everything goes well. In other cases, if the telomeres are shortened and the cells do not die, it may instead turn cancerous, which, since we're talking about a prematurely aged cell and consequential organism, you may have children developing cancers that are observed in much older people, or animals and similar. This is why Dolly the sheep died at about half the age of a normal sheep. She died at 6 years old, whereas a regular sheep would survive up to about 12. Even when clones are genetically identical with another clone, they will not be identical in physical or behavioral characteristics. This is because DNA is not the only thing that dictates this sort of development. In fact, in some cases, it's the other way around. Exposure to different stimuli, whether that's air purity, education, food, and so on, all of these will have different effects on the genome. And so, two otherwise identical clones will develop along similar, but not the same line of development. This is because each of them will experience different inputs to alter them, even in embryonic development. Because of this, they will be different at the end of their development than they are at the start. This is true no matter what environment you put them in. Even if we look back to the start of this video, where we look at scientific tropes, you'll see that no matter how they do this, there's no way that they could create that many perfect, exact replicas of a clone. This is about the closest you're going to get. Pint-sized toy models of clones. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.